been majorly geeking out this morning over cell-free protein expression, and especially the Pure system, which is protein synthesis using recombinant elements. So basically, these people, they purified out all of the different components that are required, the minimal components required to make a protein, and mix them all together. And now this is sold commercially, and it's a really powerful tool both for making proteins and for studying the translation process. And I think it's really, really interesting. And I've been to take you guys through the paper a little, and just kind of geek out a bit. So I hope you'll indulge me. Okay, so this is going to be a kind of weird video because I just kind of whip some stuff together, but I've been thinking about it and I just think it's really cool and I want to talk about it. And so when I said these people, so this was done by Yoshihiro Shimizu et al. In 2001. And this is now sold commercially and stuff, you can get it from NEB. But it's all based on this this paper, which I want to tell you a little bit about a little more about, and I'll walk you through some of the figures and that sort of thing. But the key thing to know is that it's kind of one of two basic approaches to studying to doing cell-free protein synthesis. And so normally if we want to study a protein, we take the instructions for the protein and we clone it into a plasmid and we stick that into cells and we get those cells to express it for us, inside the cells. So we can stick it in bacteria and have the bacteria make it, or we can stick it in insect cells or even mammalian cells. But in all those cases, the protein making is happening inside of cells. And so for numerous regions, this can be a bit of a pain. And so someone who studies protein and has done tons and tons and tons of purifications of liters and liters of cell lysate, you have to break open the cells and purify out your protein. And there's a bunch of other stuff. And so the idea of being able to do it outside of cells is pretty appealing. But of course it's not just that easy. So there are a couple of you can basically, the approach is just break open the cells and get rid of the membrane stuff before they make the protein. And so this is basically the lysate-based approach. And so these lysates can be bacterial, they can be wheat germ, rabbit reticulocyte, or other cell lysates. And then if there's other approach, which is what I'm going to talk to you more about, is this being reconstituting the system from purified components. So actually finding all those minimal things that are required in order to make the protein, mixing those together, and then making the protein that way. So with either of these cases, there's going to be, it's not there are disadvantages compared to doing it in cells. This isn't rapidly replacing making proteins in cells, especially if you're doing something where you need a lot of protein. You don't really hear people talking about cell-free synthesis that much. I don't know if maybe in the future, if things are really advanced, maybe you see a lot with cell-free protein synthesis in high throughput things when people are doing things on chips and that sort of thing. But as someone who's worked a lot with proteins, you know that these things can be really finicky. And so proteins might not act quite normal if you're not making them in their normal environment and that sort of thing. So one of the benefits of the lysate-based over the purified components is that these can do things well, depending on what cell type you may use, they can do things like post-translational modification, so phosphorylation, glycosylation, that sort of thing. But of course, that's depending on what cell type you use, because those modifying enzymes that are going to add those modifications, they have to be in the lysate that you're using. So if you want to have something glycosylation, or you want to have something complex things, the bacteria is not going to help you with that. And so, but if you do, you can use human cell lysates even. Rabbit reticulocyte is really popular. This is one of the first in vitro translation systems that was used. And so rabbit reticulocytes, so these rabbit blood cells. In all these cases, they typically remove the nucleic acid, so they remove the DNA and RNA, so you're not just making the stuff the cells wanted to make originally. But actually, I have a post on Hildegard Lamprom, and they actually, people did a lot of the early work studying how mRNAs work based on rabbit reticulocyte lysate. And actually, because these things are chock full of the mRNA for hemoglobin, these cells would make a lot of hemoglobin. Then she was actually able to like use this and then mix things and do cool things to make key discoveries with mRNA. So more on her and that sort of thing in that other post, but we're not talking about that now. We're talking about this. Another thing that this lysate will have is like chaperones to help folding. So basically whatever was in the cell when you break it open. And so unfortunately, there's also sometimes some things that you don't want in the cells. So you could have like nuclease contaminants, so things that are going to chew up DNA or RNA. And you can have like efficiency problems. You can have other things in there that are taking up the ATP. And so you can have other side reaction type of things that you wouldn't have if you just had the purified components. So you can imagine that you could be doing things that are using up energy but aren't making your protein. And protein making takes a lot of energy, so that's a big deal. We want to conserve all of the energy and use that energy for our protein making. We also have batch to batch variability because basically it's whatever's in the cells. And so if there are different things in different cells, different things at different times, this is going to vary. But it's easier to do this to produce a lysase and break open some cells and get rid of the membrane than it is to actually like purify out each of the individual components. But either way I'm pretty sure it's like much more expensive than if you were to do it in cells as well. It's another thing. Okay, but so from this purified components here, you're only using the minimal things required. So this is really, really good if you want to like study the translation process, or if you want to study certain components in the process. So you can add those components to test their effects. So maybe you want to see what effects something has on translation. And you can buy versions like without the ribosomes if you want to study ribosomes and ribosome modifications. And so then you put your own ribosomes in, and they also have versions like without the amino acids or the tRNAs or the release factors, various things that you can do so that you can really customize it and you can adapt it to use like non-canonical amino acids. So I'll talk more about that. They showed like a proof of concept in their original paper with this. So it's really, really good for research when you want to test individual things that you couldn't just like take out of the cell lysate. So you couldn't just easily take out the normal tRNA, like if you want to use a modified one, you'd have to be competing with the normal one if you just use the lysate. But if you use the purified components, you can stick in all of the modified one, so you can have better incorporation of your modified amino acid. And so you can actually use modified ones to actually test if it has some sort of label, then you can actually see how the measure translation and stuff. So I've seen a lot of really cool stuff done with that. But you can't you're not gonna get, your protein is probably gonna be less like normal than you would get from like having a lysate, and definitely from being in the cell, because you're not going to have those post-translational modifications unless you add something in. You might have folding problems. So like in cells and in the lysate, there's going to be chaperones. So like proteins that help your protein fold. You're not going to have those unless you put them in here. And so your protein might not fold correctly. But if you want to study translation and stuff, then this is a really cool system, which is actually how I came across it. And so in my postdoctoral research, I'm going to be doing some work with ribosomes. And so I've been reading up, and a lot of the papers, they were like mentioning, like citing this, that they use the pure system. And I'm like, what's the pure system? And they were using this pure system. More specifically, they were using like the delta ribosome version, so the one without the ribosomes. And so then they could add their own ribosomes and test things. And so that's kind of like how I came across this, because then I'm like, well, what's inside that? And so if you go to like, we'll go into the paper more, but just so you get like an overview of what's inside this, basically they have the translation factors, so the things that are needed for the protein making. You need the initiation factors, elongation factors, release factors. Don't worry, we'll go into this a little. The ribosome recycling factor, and then the things to make charge the tRNA. So to put the amino acids onto the tRNAs, you need these amino acid tRNA synthetases. this one, I cannot say that word, methionine tRNA formal transferase. So basically in bacterial synthesis, so this is used, they use like bacterial components, and in bacterial protein synthesis, the first amino acid, the methionine that starts things off, actually gets a special modification, this formal group added. And then it also has the ribosomes, the tRNAs, the energy regeneration system I'll talk to you about, and then NTPs, amino acid salt buffers. So basically that's what's inside. And we'll look more about how all of this came together and in more detail in a minute. But for those of you that can't wait, basically the Pure system contains everything that's needed to do the transcription, so to make mRNA copies of the gene, and to do the translation, to make your protein from that, as well as to regenerate the energy. And so in bacteria, you have the transcription and translation are coupled. So in our cells, we have the transcription, we have the mRNA copies being made in our nucleus, and then the protein making is actually happening in the cytoplasm, so they're uncoupled. But in bacteria, they're coupled. And so what's happening in bacteria is that the transcription's happening, the mRNA is being made, and then right away, as it's being made, it's getting translated. So you're making protein directly when you're making the mRNA. And in this Pure system, what you're doing is you're adding in the DNA instructions, and then if you add T7 RNA polymerase, and in the original paper they had T7 RNA polymerase in there, then you can couple the transcription to translation. So if you don't, you can add like mRNA, but as they found in like this paper, like mRNA can have like structural problems that can make it less efficient. But in any case, so if you go from the dRNA, which is what they did in this original paper, then you need like the RNA polymerase, which is going to make the mRNA copies. These mRNA copies are then going to be used by the ribosome to make the protein. And then in order to keep this all going, we're going to need to regenerate energy. And so there are a couple main spots that you're going to see energy used. This is a really energy expensive process. So in translation, what happens is it's really, really expensive. So first you have to spend ATP in order to charge the amino acids. So these amino acid threonine synthetases, they're going to actually use ATP to charge and like activate these amino acids so that they can get added on so that then they can get linked together by the ribosome. And then when the ribosome's doing its thing, that's going to take energy too. And so you're going to need GTP, and you're going to need ATP, and you need ATP to make the GTP. And basically you need to be regenerating the ATP and the GTP. And so your GTP is going to come in when you have like these, you're going to have it used in both like the initiation. So this is where our initiation factors are going to come in as well, why we need those. These are going to help set things up, and they require energy. Then you go through elongation, and then you go through, so these peptides are getting linked together, and then you have termination, where instead of linking a peptide, you have this release factor come in, and so that's where the release factors are needed. And these release factors are basically, instead of adding a peptide, getting the peptide to attack and link together, they get water to attack, and now the peptide chain is released. And so all of that is going, so you have these multiple steps that are going to need energy, and so we need to be able to regenerate it. And so how this is done is that they use several things. And so basically, so we need the ATP for charging the amino acids, charging the tRNAs. And so for this, they're using creatine phosphate and creatine kinase. And so creatine, I talked about this in yesterday's post, but creatine is actually something that's like used by our muscles in order to store phosphate groups that can get transferred onto ADP to make ATP by creatine kinase. And so in their system, they have creatine kinase and they have creatine. And so because when you're charging these amino acids, so when you're adding the when you're charging the tRNAs, what you're doing is you're actually, instead of spending like just one phosphate group, you're spending two. So now you need to add back on two phosphate groups. So they have to use a few different kinases. And so they have another kinase that is going to, I think it's like adenylyl kinase. I have to look at it again. But they have a kinase that is going to add add a phosphate group to AMP to make ADP. And then the creatine phosphate kinase is going to add the phosphate group to the ADP to make ATP. And so you need to use two creatine phosphates for every one, because you need to add two phosphate groups on. So that's where, like, why they say two creatine phosphate, two creatine, two ADP, two ATP. So basically, this is per tRNA, per like amino acid that you add. So here you're already spending a lot of energy to regenerate this. And so you like, but you are able to regenerate the ATP. When it comes to the GTP, here you have the, you're using ATP to make GTP from GDP. And this is done by a nucleotide diphosphate kinase, which is basically, it's able to take the phosphate group from ATP and put it onto GDP to make GTP. And then that can be used by all of these translation factors that need it. You also see this like N10 folinic acid. So basically this is needed. This is where like the formal tRNA formal transferase is coming in. And so this is going to add that, that funky group onto the initiator tRNA methionine. So you have basically, you're regenerating your energy, and then you're also transcribing and translating. And so all of this is inside of this really cool system. And so what's really, really cool about the system too, is that when they're doing their purification, which I'll go into it more in a minute, because I just like talking through papers, they his tag the protein. So his tag, basically they stuck this chain of histidines on the end, and this is going to allow it to bind to a nickel affinity column. So if you have this little like column filled with this resin, these little beads, and they have nickel on them, that's going to stick to the his tag. And then you can wash things off, and then you elute off. So you like compete off with like imidazole, which looks like histidine, but it's not. You can then push off your protein. What's really cool, so normally we use that in order to like purify proteins, and that's how they use it in order to purify the various components that they express. So all of those different tRNA synthetases, all of those various elongation factors, initiation factors, they have his tags on them, and then they can do this purification to purify those out, to purify those proteins. But they leave the tag on. So normally we take the tag off when we're doing like structural biology or biochemistry stuff. Like we don't want the tag there to interfere. But here they can leave the tag on, and then they can use reverse nickel affinity chromatography later, which is cool. So basically later, if you want to think about it, you want to remove those, because now the protein you want is basically the only protein that's in the mixture that's not tagged, except for the proteins in the ribosomes, which they are going to get rid of a different way. So basically they use the cellular ribosomes, and so they purified these out of E. coli, but just like the native proteins, there are ways you can do this with like sucrose gradient and that sort of thing, because these ribosomes are really, really big. And so they're going to use that bigness of the ribosome in order to get rid of them after the reaction. So after the reaction, they basically take their product and they put it through, so they have this mixture, that mixture with the defined components, as well as now their protein that they made. And what they're going to do is they're going to put it through, use ultrafiltration. So basically we use like ultrafiltration when we're like concentrating a protein and that sort of thing, because they have these membranes with these holes, and you choose like a size hole that's smaller than your protein. And so your protein is going to stay in the little copy part on top, and then your other stuff, all the rest is going to flow through. And so when we're doing like concentration like that, that's we want the stuff that stays in the cap. But here, this is like the opposite. They're saying we want to capture the really big stuff up here and get rid of it, and then use this stuff that flow through. So they take this stuff that flows through, the filtrate. So basically, they use a membrane that the ribosome is too big to get through. So the pores in this membrane are too small. The ribosome can't get through, but your protein can. And so the protein is going to flow through. And then, but you're all the protein. So you're also going to have like the proteins from, you're going to have like all those proteins that were part of the mixture from the pure system. So you want to get rid of them. And so you can get rid of them because you have that tag. And so now this case, instead of washing things off and then pushing off the protein you want, basically you have a nickel column, and your nickel is going to get stuck, and then your other stuff is going to flow through. And so the things that flow through is the stuff you want in this case. And so your proteins get through, and the tag stuff gets stuck. So you can see here from the paper, so this was their reaction mixture. So you can see that in the pure system, there's a ton of various proteins. This is like an SDS page gel. And so you're looking at stained with lipomassy. So you're seeing all the proteins, and you can see that there's a lot of them because you have all those ribosomal proteins, you have all those initiation factors, elongation factors, all of those various things, the enzymes needed for the tRNA charging. But then when they flow this through, now you can see that they have their product is staying, but all that other stuff is gone. So it's really, really cool. So let's look a little closer at the paper. Okay, so this paper was published in 2001 in Nature Biotechnology. And as you can see by the citations, it's been used a lot. So basically, they say that they got about like 160 micrograms per mil per hour in batch mode. And so that, and let me see where there was in this, there's a review paper or news and views that went with it, the one where I got this paper from. So they said that there were some cool stats in here, which I'm guessing were in the other paper too. It had a synthetic rate of about 1.8 peptide bonds per ribosome per second, compared with approximately 20 in vivo. And so they're like, yeah, this is relatively slow compared to what happens in cells. But in my head, I'm like, whoa, what happens in cells is super, super fast. And so is what happens in here, 1.8 peptide bonds per second, because that's a lot of work going on. But anyway, let's go back to the paper. And so here you can see that, so in their paper, what they did is basically, so they have the initiation factors. So there are 3 of them. So this is all in the E. coli system. Things are different in like eukaryotes and that sort of thing, but this is all involving the E. coli system, which is a little simpler to understand, thankfully. And so in E. coli, there's 3 initiation factors. So those are required to help get things set up at the in that initiation stage, get the ribosome to the right place, get that initiation tRNA there, all of that stuff. Things get more tricky with the release factors. So there's these 3 release factors, RF1, 2, and 3, as well as RRF for termination. So I had to look a little more into this, but like RF1 and RF2 can apparently act as, they can recognize kind of different codons. So you can use one and not the other in some cases. And so they take advantage of that in order to incorporate unnatural amino acids, as I'll show you. And then RF3 is going to help kind of release them after they do their stuff with the help of RRF. Which is so RRF and RF3 are going to help with recycling of the ribosome. So helping everything kind of reset so you can have like multiple turnover. That was from this paper. So Graf et al. 2018. But going back to this, I just wanted to, because that kind of confused me. So because they say like RF2 is not required for the translation of genes terminating with these codons. So they actually don't have RF2 in like their initial mix, but I think that based on the NAB one now it is. I'm not exactly sure. But okay, so they had to purify 32 components individually. So they purified the initiation factors, the elongation factors, those release factors, this ribosomal recycling factor, and then 20 amino acid tRNA synthetases. So the things that add the that are going to help with putting the phosphate, putting the amino acid onto the tRNA so that the ribosome can use it. This methionine tRNA transformylase, which is going to make that modified methionine for the initiation. T7 RNA polymerase, which is going to make our mRNA copies from the DNA, and the ribosomes. But these ribosomes are not tagged. These are just like the native E. coli ribosomes. Then they just buy a mix of tRNAs and TPs. So like your ribonucleotides and your deoxyribonucleotide triphosphates, then creatine phosphate, and which was needed for that energy regeneration cycle. 10 formal, this thingamabob for that formal group for the, the initiator amino acid. 20 amino acids, again, just things that they bought. Creatine kinase, myokinase. So creatine kinase is the one that is used to regenerate the creatine phosphate. Myokinase, so this is the one I think that is used to put the second, the ATP, the phosphate group onto the AMP to make ADP. Nucleoside diphosphate kinase, which is needed to make the GTP, and then pyrophosphatase. And so pyrophosphatase, apparently this, if you have this, so when you have this during the charging step, you make AMP plus pyrophosphatase, sorry, it's a really hard word to say, which has two phosphate groups linked together. And so this apparently is really good at sequestering magnesium. And so magnesium is positively charged. These pyrophosphate groups are negatively charged, negative, positive. You can get this magnesium hidden, which would not be good if you need that magnesium to be working for other reactions. And so you can add pyrophosphatase. And so what it's going to do is it's going to hydrolyze the PPI to phosphate. So basically break that two phosphate groups into individual phosphate groups, and those are less good at sequestering the magnesium. And so then you're not going to have your magnesium be hidden. And so that's why they have that. So they do their purification of these factors, which is like a lot of different factors. And so basically they made them as his tag fusions, and so that they have that his tag on the end. And then they use an imidazole gradient. So we talked about with the his tag purification, how you can compete it off with the imidazole. And so that's what they did. And these are their purified enzymes. I do not know how they have a gel that is that wide. That is impressive. But they have, they purified all of these components. And so you can see the initiation, elongation, release factors, and all of the different tRNA amino acid synthetases. Then what they wanted to do is they wanted to like make sure everything's pure and see what's actually really needed and that sort of thing. So they purified all these components. Then they tested to make sure that like there weren't things, both to see what was absolutely needed and to make sure that there actually don't have other contaminating things in there. So like if they get rid of something they know is like really important for this, then they're not going to see the activity. So if they like get rid of one of the tRNAs, amino acid synthetases, then they're not getting then you're not going to have the protein made because you're not going to be able to add that amino acid. And so if there was some contaminant in there that could do it instead, then you would see activity and it'd be like, uh-oh, we've got a problem. But everything seemed to be working properly. So that was all good. They also did things to evaluate the release factors that I'm not going to go into. But yeah, so they found that the ribosomal release factor increased the amount of peptide and gave the multiple turnover because that's helping recycling with the recycling of the ribosome. And yeah, so then they tested like the individual activities. So this is where things kind of got really confusing, and I got a little down a rabbit hole trying to look into RF1, RF3, RRF. But basically they used this assay where they had, they used like an amino acid, this synthetic mRNA with this like short sequence for this peptide. And then they mixed it. So they used that like the methionine tRNA methionine. They used it that was like radio labeled. And then they mixed it with this mixture with like different ones of these factors, and then measured the amount of the peptide that was made by like seeing how much was like of the radioactive stuff was like used up and that sort of thing. But so that was how they tested that. And then they wanted to see, okay, so we're making peptides, but are we actually making proteins? And so they had, they tested the ability. So now they have genes for these different proteins. So they have this DHFR, this lysozyme, GFP, GST, T7, G10. Not exactly sure what those are, but they used radio labeled methionine, so they could then see really tiny amounts of protein. So radioactivity is really sensitive, and so they do these in vitro translation reactions. And then they see that they're making protein, although they didn't have very good efficiency with the GST. And they saw that the protein that they were making was active. And so they measure this like enzymatic activity of DHFR, the bacteriolytic ability of this lysozyme, and GFP fluorescence, and they saw that they were all active. And then they did like, they wanted to see like why was things like, how much energy, how efficient was it like running out of energy? And so basically they did a time course of the synthesis of DHFR at different ribosome concentrations, and in all of these cases they saw that they were running out of steam. So at about an hour they started kind of leveling off in their production. So they wanted to see if this was like, was it running out of energy? So basically they say that they tested this. So they looked at the amount of energy that was available. And they found that so this S30 system, this is like a bacterial lysate. And so this was trailing off really fast, whereas their system was still holding up, but you can see it's going down. And so this is one of the advantages we talked about with this pure system, is that you don't have all of those like side reactions potentially happening. So you can have higher efficiency in your conversion, but you're still like running out. And so that's something that was like worked on afterwards. And they said like, we need to work on this, people need to work on this. And I think they did. But I still don't know how great it is exactly. But then this is just talking about what we talked about with the purification of purifying the stuff out. And then finally, I think, so this is like the incorporation of unnatural amino acids. And so I talked a little bit more about unnatural amino acids in a post a few weeks ago, I think, where basically you can replace like one of what's normally a stop codon, change it to use a tRNA that actually is going to sneak in a non-canonical amino acid. And so in this case, they suppressed the amber codon. So this UAG, they substituted it for the, basically, because you have those ribosome, the release factors, there's like those different ones that recognize different things. And so because they only need to recognize like they only need to recognize the stop codon. And so if they remove that one and then they change the like stop codon to the other one, and then it can still stop where it's supposed to. And then if there is one of these, they put in one of these like hidden stop codons type of things, then you can, and you don't have the stop factor for that. So it normally would be a stop, but now you don't have that release factor. So now you can sneak in an amino acid, a non-canonical one. So that's what they did here. Except that here they were basically, using it as a model for unnatural amino acid incorporation. So basically this was just like a normal amino acid, it's just like a valine. But they were able to, so now they're looking at, okay, does it make a truncated product or does it make a full product? You can only make a full product if you have the if it's not stopping at that stop codon. And so because they substituted it so that the stop code, what was the stop codon is now spelling valine, now they're going to get a full length product, and that's what they see here. But the real excitement is like the potential it gives to do all sorts of really cool stuff with non-canonical amino acids, as well as because you're putting in the tRNAs, you can put in tRNAs with various modifications just to replace them all, kind of thing. And yeah, so that's the basic things. This paper, like there was a lot of work involved, lots of things purified, and yeah, so it's just really cool, and it's a really great tool for people who want to be studying things in more detail, like what's going on in these reactions, what happens if we add things, what happens if we do that sort of thing, as well as doing cool modifications. So cool stuff to keep an eye on, and hopefully you enjoyed geeking out with this, because I think it was really cool.